Good morning, class 11th children. In biology, we will continue with kingdom Monera, and you see that in the last video, last to last video, number 5, we studied about the generalized transduction, specialized transduction, which are forms of sexual reproduction. We saw transformation in sexual reproduction of bacteria. We also saw the economic importance of bacteria, harmful pathogenic activities of bacteria, food spoilage, water pollution, and in video 6 we also study about the useful activities of bacteria like sewage disposal, use of bacteria in medicine, use of bacteria in energy production. Household products were prepared by bacteria like yogurt, cheese, curd, etc. Then increase in fertility, importance in industry, dairy industry products, we saw the role of bacteria. Then dextran productions, we saw the role of bacteria. Production of acetone and butanol like compounds, chemical compounds with the help of bacteria. Curing of tobacco and tea to give flavor to these products with the help of bacteria. Decomposition of dead organisms, microbial control of insects with the help of bacteria. Retting process, where we saw flax and other fibers, they were purified so that they could be taken out for usage for making mats and baskets, etc. Then we also saw in usefulness of bacteria the degradation of petroleum, hydrocarbons, and bacteria in genetic engineering. We further see that in genetic engineering, children, the bacteria can be put to several uses. For example, if you see here in your book, page number 78, we see several applications in genetic engineering of the bacteria, like Bacillus thuringiensis. It is good for production of Bt toxin, which is a highly potent, safe, biodegradable insecticide for plant protection. Then we see Escherichia coli, which is a bacteria which is applied for the production of human insulin and human growth factor interference. We will study the details of this in class 12. The chapter would be in great detail in class 12. Then we come to Pseudomonas fluorescens, which shows an application in prevention of frost damage to plants. Then we see bacteria like Pseudomonas putida, which is a scavenger of oil spills. Then we see Rhizobium melliloti, which is another bacteria which applies into nitrogen fixation in the cereal crops in the form of leguminous nodules. Now today we come on to Mycoplasma. If you see here, Mycoplasma or PPLO, they are the smallest known living cells without a cell wall. As you can see here, they are without a cell wall. These are the smallest known living cells, and these microorganisms were first discovered by a scientist called Pasteur in 1843. He discovered these cells while studying the agent which causes pleuropneumonia in cattle, like buffaloes and cows and bulls. He found that pleuropneumonia disease was being caused there. So they were designated as PPLO, or the full form of PPLO, which is written here, is pleuropneumonia like organisms. Pleuro, P for pleuro, pneumonia, also P is silent, so this is pleuropneumonia, and L is for like, O is for organisms. They are contaminants in the organic matter. They are also found in hot water springs and other thermal environments. They are found in hot water springs. Then they occur in soil. They occur in sewage water, in drains. And these microorganisms or mycoplasma, they have different substrates in humans. Also in animals and plants, they are spread out. Then we see the structure of mycoplasma. They can pass through many filters due to their plastic nature. They can pass through very thin areas, a very thin filtering apparatuses. They grow on media which do not contain live tissues. They are intermediate between bacteria and viruses. Mycoplasma are considered to have a structure which is intermediate between plasma and viruses. You can see here they show pleuromorphism, or they occur in nature in various forms, like these can be cocoid forms, granular forms, they can be filamentous, they can be cluster-like. And the cytoplasm, it contains ribosomes, and it also contains nucleoplasm like structure, and the genetic material is composed of both the DNA and also the RNA. Contains both DNA and RNA. We see they mostly occur in other prokaryotes, we see, and is perhaps the lowest limit required for a cellular organism. This is the smallest, the lowest limit, having DNA, RNA both, and also ribosomes in it. The mycoplasma are gram negative and stain very slowly on long exposure to colored dyes. Usually they are non-motile, that means they do not move from one place to other. Some forms, however, show gliding movements. They glide from one place to the other. And the reproduction is by budding. They reproduce by budding or by binary fission, which we have already studied in the previous videos. The mycoplasma has also been known for causing diseases. They cause serious diseases in human beings, animals and plants. They affect the respiratory organs like the central nervous system in humans and animals, the cardiovascular system of the heart, and the veins and arteries. It also affects the urinogenital system, the kidneys, and all the other parts connected with the kidneys of human beings. So they are very, very dangerous. Then they cause pleuropneumonia in cattle, that we have already studied. Pleuropneumonia causing organisms are the mycoplasmas. Then we see some of the leaf hopper transmitted diseases showing yellowing of the leaves, and witch's broom, which is symptomatic in plants and are due to mycoplasmas. They make the plant look like a broom. So we see how mycoplasmas, they are economically very important. Then we come to another set of fungus which is known as actinomycetes. The actinomycetes, or where actis means ray-like, which has got ray-like structures coming out of it, and we see mycis. Mycis is a name given to fungus. So actinomyces means ray-like fungus. The fungus are bacteria with cylindrical cells. Can you see here? Cylindrical cells, which are usually united to form filamentous mycelium. You see here the mycelium, a filamentous mycelium of a true fungus. In actinomycetes, as in true bacteria, there are no well-defined nuclei, but many chromatin granules are present in most of the ascomycetes are non-motile and they are gram positive in character. They generally multiply by means of fragmentation. Some species also reproduce by means of spores. We see a sporophoretic species, so they multiply by means of spores, or they reproduce by means of conidia, by breaking off certain parts called conidiophores. They multiply. Then we see ascomycetes are strictly aerobic and occur abundantly in soil. They occur in water, they occur in mud, they occur in manures, and in milk also, in other food products also. The ascomycetes or the fungus, they have been found by scientists. Then another property of actinomycetes we see is that most of the actinomycetes are saprophytes, but some are parasites. Saprophytes means that they thrive on dead organic matter, which is dead. Parasites are those which cause serious diseases in plants and animals. There are some useful actinomycetes which are useful. They are capable of synthesizing antibiotic substances. We know about many antibiotics, children, which we have been hearing about right from our childhood, like streptomycin, neomycin, terramycin, chloromycetin, all etc. All others are produced by different fungus or actinomycetes. These are beneficial to man in curing infectious diseases. Then we see in plants, Frankia is an actinomycetes. Frankia is a nitrogen fixing actinomycetes which helps in fixing nitrogen in plants. So children, we will study from here further about other kinds of microorganisms. Here we would finish today's study. Wish you all the best. Have a good day. Thank you.